Different demographic groups display a range of responses to drugs, highlighting the necessity for tailored considerations for populations such as children or those with underlying medical conditions. This variability significantly influences trial design. The safety of trial participants stands as a primary concern during the early phases of drug development, mandating vigilant monitoring for potential adverse effects. Phase 1 trials specifically emphasize safety and tolerability. Toxicities, whether predictable or unpredictable, can arise at any point during drug development, underscoring the importance of creating extensive safety databases prior to marketing. Notably, certain reactions may only become apparent post-marketing. A case of myelosuppression was reported in animals following the approval of a product, prompting an update in labeling to include necessary warnings. This situation underscores the importance of continuous post-market surveillance. Selecting the maximum recommended starting dose, or MRSD, is critical to mitigate toxicity while still achieving trial objectives. This process employs an algorithmic approach informed by data from prior animal studies. There's a stark variability in drug responses between animals and humans, necessitating careful translation when adjusting doses from animal models to human subjects. Understanding these disparities is essential to avoid unforeseen toxicities. Establishing an appropriate starting dose is crucial, as it can greatly influence the safety and efficacy of the investigational product during the trial. This involves thorough evaluation of all available data. Vigilant monitoring and observation of participants post-dosing are vital for early identification of adverse reactions, which helps to safeguard participant safety and shape future dosing decisions. Different drugs may necessitate varying observation times. Incorporating explicit stopping rules within the trial protocol is essential for swiftly addressing unacceptable toxicity levels. This precaution ensures that the trial can be promptly halted if serious adverse effects are detected. Various sources of safety information, including non-clinical data from in vitro studies and animal research, play a critical role in evaluating a product's safety profile. These findings enhance our comprehension of the associated risks. Clinical trial data is indispensable in safety evaluation, as it reveals information about adverse events specific to the patient population. This knowledge informs decision-making regarding the product's overall safety. Post-marketing experiences and published medical literature provide valuable insights for safety monitoring, shedding light on real-world effects following a product's release. This data can help in identifying long-term safety issues. Causality assessments are vital in determining whether an adverse event is attributable to the study drug. This understanding aids in making informed decisions that prioritize patient safety. Case studies exemplify how unique scenarios, such as anaphylaxis triggered by a drug, demand immediate reporting to sponsors. This underscores the critical need for vigilance throughout clinical trials. Unexpected adverse events require prompt reporting if they are not cataloged in the investigator brochure or display significant differences in severity. Accurate classification is imperative for effective monitoring of drug safety. Distinguishing between suspected and established adverse reactions is crucial, as this distinction impacts regulatory reporting and the comprehensive evaluation of drug safety. The FDA mandates that 3 specific criteria must be fulfilled to submit an IND safety report, particularly concerning serious and unexpected events with causal evidence linking the drug to the adverse event. This standard is designated as SUSAR. Safety reports must be submitted to the FDA within prescribed timeframes, with the most severe cases requiring reporting within 7 days. This emphasizes the urgency of monitoring adverse reactions. Legislation surrounding pediatric drug development, such as the BPCA and PRIA, was established to improve children's access to approved medications. These laws not only mandate studies but also incentivize drug developers. Ethical considerations hold paramount importance in pediatric clinical trials, ensuring that children participate only if there is a prospect of direct benefit. This safeguards their rights and well-being during research. Special formulation requirements are vital for pediatric medications, as children may need distinct strengths or forms to guarantee safety and accurate dosing. This takes into account their unique developmental stages. A multidisciplinary approach is essential in pediatric drug development, involving experts such as pediatric specialists, clinical pharmacologists, and biostatisticians. Their collective expertise ensures thorough evaluation tailored to pediatric needs. The Pediatric Research Equity Act, or PREA, requires early submission of pediatric study plans to the FDA, facilitating guidance on pediatric therapeutics while ensuring drug safety for young patients. This requirement aims to streamline drug approval for pediatric use. Historically, pregnant individuals have often been excluded from clinical trials. However, recent trends indicate a shift towards their inclusion. This change is imperative for collecting relevant safety and dosing information for pregnant and lactating populations. The FDA's draft guidance issued in 2017 outlines critical considerations for the safe inclusion of pregnant individuals in clinical trials. This guidance ensures that potential risks and benefits are adequately evaluated prior to enrollment. Recent reports reveal a surprising deficiency in evidence concerning liability in the inclusion of pregnant individuals in trials. This suggests a pressing need to alter perceptions surrounding their participation in clinical research. Efforts to enhance diversity in clinical trials are essential for the well-being of public health. The FDA seeks to include underrepresented populations, ensuring that drugs are both safe and efficacious for all demographics. Understanding diversity within clinical trials is vital for accurately reflecting the demographics of drug users, particularly among vulnerable groups such as pregnant women and children. This inclusivity can lead to improved health outcomes. Investigators play a crucial role in monitoring patient safety during trials, especially concerning the assessment and reporting of adverse events. Their insights are invaluable for comprehension of drug impacts across diverse populations. The ethical considerations surrounding pregnancy during clinical trials present significant concerns regarding patient safety and follow-up protocols. Best practices include reconsenting patients and collecting critical pharmacokinetic data for effective oversight. Recruiting diverse populations for drug studies is paramount for ensuring the effectiveness of medications across various demographics. This effort fosters trust within historically underserved communities. The critical importance of mortality as an endpoint in clinical trials targeting serious illnesses cannot be overstated. It serves as a fundamental measure of a drug's efficacy and safety. Extrapolating dosages for neonatal populations demands meticulous consideration of their distinct physiological differences. This ensures that treatments are administered safely and effectively to younger patients. Neonates possess unique physiological characteristics that differ significantly from older children and adults, necessitating specialized study designs to achieve accurate drug evaluation. Utilizing existing data from older pediatric populations can effectively inform dosing and safety assessments for neonates, potentially minimizing the need for extensive new trials. Innovative trial designs, such as Bayesian approaches, can be advantageous in tackling the complexities inherent in pediatric drug trials, particularly concerning rare diseases. The Investigator's Brochure, or IB, serves as a crucial resource for informing clinical trial investigators about drug safety, formulation, and pharmacological effects. It must comprehensively include key information derived from prior studies. Reporting adverse events is imperative in clinical trials. Investigators are tasked with clearly communicating the causes of events, such as death or anaphylaxis, to ensure comprehensive understanding and accountability. Upcoming discussions will focus on non-clinical and early clinical drug development, including key areas such as pharmacology and toxicology. These foundational elements are critical for the design of effective clinical trials.